AMS, FACBI, FRC, Pathology, Chairman and Senior Consultant, Department of Biochemistry, Sir Gangaram Hospital. Thank you very much, ma'am, for accepting our request. The subject today is the paradigms and paradoxes of sigma metrics in medical diagnostic laboratory. So the testing in the medical laboratory is a very important part. And I think that in India, most of the medical laboratories are not accredited. Very few medical laboratories are accredited. So basically the testing in a medical lab is very important. On the basis of that, the doctors prescribe what action has to be taken. We have with us Professor Seema Bhargav, who is basically having a chairman in senior consultant in Department of Biochemistry at Sir Gangaram Hospital. She is the president of Association of Clinical Biochemists of India. She is a member of International Federation of Clinical Chemistry, Evidence-Based Laboratory Medicine Committee, and Visiting Lecture Program. She is a chairman of Diagnostic Division Consortium of Accredited Healthcare Organizations. Ma'am, we are actually thankful to you. You have accepted our request and ready to deliver a talk, which is very important for our large number of participants, laboratories, etc., how to make the testing, how to develop the testing, and how we can help them. Your experience, your research, your awards, your publications. If I will start reading, it will take 2 hours to read, and your time will be, I will take your whole time. So I'm not going to read or going to tell each and everything. But everybody knows, being over there in Gangaram Hospital, we can understand how much pressure, how much work responsibilities you must be sharing. And it is obviously for the noble cause. I think the doctor profession is the noble profession where you can actually serve the society to the patient actually who needs your help. We have with us our Honorable Director Sahab, Shri B.N. Dixit Sahab. Sir, would you like to speak 2 words, please?
So I welcome Dr. Saab, that she has spared her some times to provide her thoughts and knowledge to we people. Legal metrology is very large topic. Discover all type of measuring equipment and measuring values and measurement of uncertainties and type of measurement and all the things are covered. So it is covered from the medical point of view because there also the measurement things are there. So hospitals are also may take the facility of the RRSLs, and madams knows very well that what is the important of the measurement in the laboratories. So I wish and pray for the success of this seminar. Thank you all. Thank you, Dr. Saab.
Thank you very much.
Thank you very much. Yes, ma'am. Please. Now you take over, please.
I'm very humbled by the warm welcome and the very kind introduction by Mr. Agarwal and Mr. Dixit. Thank you so much. I have to thank Mr. Ashutosh Agarwal as well as Professor Venkatesh Tupil, who is the reason I'm here today, because he is the one who suggested, and it just happened. I am honored to be on this platform, actually. And I hope I do justice to it. I am a medical by profession, I'm a doctor by profession, and I have learned about sigma metrics on my own, and I have not done any course for it. And I'm not a statistician, but through simple calculations, I hope to take you to a level where you will understand what I am thinking. I am just putting across my thoughts about it, what is good about it, what may not be so good about it, where we need to be very careful. And at the outset, though I'm talking with respect to a medical diagnostic laboratory, the very same holds true for all laboratories where, say, our foods are tested and certification is given, the FSSI mark for each food. And we are talking all the time about how the consumer must be aware. So I hope I do justice to this, and I hope I help everyone, and I hope I also gain some experience with you. So with that introduction, let me start my presentation. The topic I'm addressing today is the paradigms and paradoxes of sigma metrics in medical diagnostic laboratories. Okay, my slide is not. Can you stop using my thing? Because I can't forward my slide now. Is there any way that I can do that? Can I get some help?
Santosh ji, what help can we do now? Ma'am, I think she's not able to move in the slides. Next slide.
So do I decline the request? Let me decline it and let me try on my
Please.
own.
Please.
No, okay,
Rather than
I'll
you kindly
approve
share
it.
it to
So
me.
could you
kindly share it to
please?
me, WhatsApp or
Yes.
mail. We will share from this side.
Yes, it's come.
Okay, good.
So the thing that sorry, go back. So what I'll be addressing today, I will start with a short introduction. Then I'll talk about Sigma and quality and how did it all start. Just a short brief history, just for those people, for the benefit of those people who are probably not medical and who probably are not statisticians either. But it'll help them to understand the topic. And then I'll get on to what exactly is Sigma Metrics and Six Sigma. Then what is its role in quality assurance of a medical diagnostic laboratory? And that would be the paradigms. What are its limitations or paradoxes? So starting with the introduction, I'd like to, all of you are very well aware that this industry, in every industry, whether it is a car industry, whether it is a mobile phone manufacturing industry, or whether it manufactures these tubes that are used for sample collection of patients, that is called the blood collection tubes. All these establishments are committed to making continuous efforts towards achieving stable and predictable process results, especially in those processes where the characteristics of the process can be defined, measured, analyzed, improved, and controlled. And this is the acronym DMAIC, which we will be using later also to some extent. So what applying Sigma metrics, the aim is to reduce the process cycle time, to reduce the cost, to reduce defects, to increase accuracy, increase capacity and throughput, reduce pollution. See, every company is actually throwing out into the environment something or the other. And through Six Sigma processes, we can reduce that also. Then increase customer satisfaction. Ultimately, the customer is the Lord, and he will govern what is acceptable and what is not. And I'll come to why that is so. And it also may help you increase profits. So this is the define, measure, analyze, improve, and control. Basically, for define, you have to identify which problem you're going to address and what your objectives are. So then you measure what you need to be able to characterize that problem or objective. You analyze the whole process and see where there is likelihood of some risk. You do a risk analysis. And once you have analyzed and identified the possible risk or the possible area of improvement, you implement an improvement, and ultimately you control that process and make sure that the improvement implemented is consistent and is sustained. So let me tell you a little bit about Sigma and quality and how it all started, and just a brief history about it. So what is the principle on which Sigma metrics is based? In 2018, Pontus wrote in his publication that if there were only one law of probability to be known, this would be the normal distribution or the Gaussian distribution or the Laplace-Gauss distribution. So with that in mind, let's tell you how it all started. In the 18th century, there was a French statistician who interestingly was a consultant to gamblers. And they actually were actually they used his services much more than probably his own fraternity did. His name was Abraham de Moivre, and excuse me for the pronunciations, French people usually have many silent alphabets in their names. I hope I'm pronouncing it correct. So he was the first to observe that even though most natural phenomena appear irregular and unpredictable, but when they are considered in large numbers, they have a very normal and predictable pattern. So from irregular and unpredictable, they became normal and predictable. And that is the basis of the Gaussian curve. And here you can see three curves that are normal, the yellow, the red, and the blue, but they have different precision limits, while the one in green is skewed. But all of them still are Gaussian curves. Just giving you an example of the Gaussian curve. So in 1718, Abraham de Moivre he wrote a book that was called The Doctrine of Chances. And believe me, who used it? The gamblers. They used it to predict how they can win. We still in the scientific world had not learned to use his principles. Then he observed that there are different phenomena have different skewness. So you may have a normal curve, you may have a positively skewed curve, or you may have a negatively skewed curve. He added this knowledge to the observation of Galileo in the 17th century that smaller errors were more common than larger errors. And the result was another doctrine, the doctrine of odds. Then in 1809, now we come to the actual person, Gauss, who gave the law of normal distribution interestingly, this was for analyzing astronomical data derived from astronomical experiments. And in 1810, Laplace gave the Laplace central limit theorem for this normal distribution. And what he said is that all errors are equally distributed around an arithmetic mean. On basis of these, he then later gave the analytical theory of probability. So what is the Gaussian Laplace or the Bell distribution? It's called Bell distribution because it is bell-shaped. What is important to note is that even though it's bell-shaped, the two ends on the plus and the minus side, the right and the left, actually never meet the baseline. They go on up to infinity. And that is why when we talk about these errors, we have this. So what we have is that if it's a one standard deviation plus and minus, you are covering 68.26 data points. And similarly, it increases with 2, with 3, with 4. And when you come to 6, it is 99.999998%. That sounds a lot, but I will show you how that could be also not enough. So when did quality come into the picture? Joseph M. Juran is known as the father of quality, and in his early 20s, he said he gave the continuous improvement as a business strategy. And it is, he said that you can never be too busy to improve because the whole ethos about any process is continuous improvement. Today we know this much and we improve our quality to the extent that we have knowledge today. Tomorrow knowledge changes, you add that knowledge and you improve further. And this is an ongoing process. In the 1930s, Frederick Winslow Taylor wrote the book Principles of Scientific Management. This was the first time there was any book about quality. And Walter Shewhart at the same time wrote Principles for Quality Assurance and Quality Control in the Engineering Sector. Shewhart's colleague Edward Deming applied it to the automotive industry following World War II. And all of you know that even though Shewhart was the origin, he gave the PDCA cycle, he was the originator of it. It is Edward Deming that is actually credited with it, and it is named after him. It is called the Deming PDCA cycle. And then there was no stopping, of course. Quality and sigma came together. There was a period of evolution. It was still, now this was, I was talking about the 1930s and 1940s, but it was not till late 1970s that the industry actually applied these quality and sigma principles to their own production lines. And in the next 20 to 25 years, there was no turning back. Every industry took it on themselves to apply Sigma metrics, do Sigma analysis, improve their processes. And I'm sure the journey has been very good. And now, in the last 3 decades or 2.5 decades, the medical industry has also applied Sigma analysis to their processes wherever possible. So, therefore, there was a marriage of quality and Sigma. So, Sigma is the ultimate marriage of experience and vision, the ability of a team to translate experience and knowledge and represent its ultimate into actions, represents its ultimate value. This was given by Douglas France, and I think this is a very apt description of what actually happens, how Sigma and quality are married. So now again, I've just given you a brief history. Now I'll tell you what exactly is Sigma metrics and what is Six Sigma. So what does Six Sigma quantify? I've already told you about the Gaussian curve, and what I'd like to say is that it is just a statistical tool for assessment of quality. Just as we know that the Gaussian curve neither end ever meet the baseline, similarly we know therefore that 100% perfection is too ideal to achieve. And going back to the curve, we see that 99.99998% of perfection is achievable, which is fantastic actually, if you look at it. And this translates into less than 3.4 defects per million opportunities. For example, if there are a million chances for an error occurring, if there are a million constituents of a process, then out of them, less than 3.4 may be defective. And that would constitute a Six Sigma process. So coming to the numerical definition, these are the definitions given by the industry, by the manufacturing industry. So that is why there is a quality grade and a yield in it. And as you can see, they also graded it as per what is its value in the thing, not just a numerical value, but what is its value in practicality. So 1 Sigma, of course, is not competitive at all. 2 Sigma is below industry average. 3 and 4 are industry average, just about acceptable. 5 is above industry average, and 6 is world class. So having given you those numerical as well as so as you can see, in 1 Sigma, you'd have 6,90,000 defects per million opportunities. Now that's quite a lot, and you don't want to have that. And coming from 1 Sigma down to 6 Sigma, you see how it drastically reduces to 3.4 defects per million opportunity. So what is Sigma metrics, and where do we get this terminology? We are talking about defects, and defects can be talked of as errors. So the International Organization for Standardization of Terminology calls them non-conformances. But in routine laboratory jargon, we call them defects or errors or false positives or false negatives or outliers. The application of Sigma metrics quantifies these defects or errors, and they enable us to identify where corrective measures can be taken. They may not give us the solution to which corrective measures to take, but they can give us the idea of where to put a corrective measure. And this, therefore, helps in minimizing the defects. Again, let us talk about the definition of Six Sigma. It is a measure of customer tolerance. Now, again, as you can see, this goes back to the industry because they are talking about the customer. Of course, today, even in the medical field, we talk about the customer. But here, it is a measure of customer tolerance. And tolerance can be defined as the amount of errors or defects that can be tolerated by the customer or the user for a given product. Obviously, for different products, the tolerance will be different, and therefore the achievement of any Sigma metrics will also be different. The tolerance limit, now till now we're talking about tolerance, and now we come to the tolerance limit. This is determined by the customer or the users and not by the manufacturer or the production system or the medical diagnostic laboratory. It is the doctors or the patients, and how this defect may affect them in their daily routine practice and in the patient's health. That is what determines what is the tolerance limit, not what I want to do or what the manufacturers want to do. So Sigma metrics standardizes that tolerance measure by measuring it as opposed to the inherent or achieved variation in the process of the manufacture of that product. Now, you know that in any process, whether it is water flowing in a stream or whether it is water in your house or whether it is electricity achievement, every process, even whether it's cooking, every process has inherent variations. And there are some that are achieved variations, which go beyond the ones that are inherent. And what we try to do is that we try to reduce in the achieved variation while accepting the inherent variations. And this happens even in the production of a car, in the production of a cell phone, in anything. So then how do we calculate sigma metrics? So therefore it is a ratio between the process tolerance and the process variance. In a medical diagnostic laboratory, what is the process tolerance? It is the process total error minus the bias. And the process variance is given by the coefficient of variation, so that we have a formula for medical diagnostic laboratories that says process sigma is equal to, in the numerator we have process tolerance minus bias, the difference between the two, divided by the coefficient of variation. So what it actually does, it calculates variations in the performance of any process. And this can practically happen all the time. And these are due to natural sources of variation as well as due to unexpected errors, what we call as random errors or even systematic errors. But they do occur over and above the natural sources of variation and over and above the inherent variations in the process. So this, so Six Sigma therefore signifies something different under changing circumstances, because as you can see, there is always some source of variation. So what it does is it takes into consideration a shift of 1.5 standard deviations in the mean performance. Therefore, we can say that a process that shows a 6 sigma performance is one that would experience up to 3.4 defects per million opportunities, even if the performance shifts by 1.5 standard deviations. But without a shift, it would indicate only 2 defects per million. So actually, Sigma metrics assesses the long-term capability or the long-term, the long-term quality of a process. So now what is the role in a medical diagnostic laboratory? So these are the paradigms that I'm talking about. Every lab wants to have accurate results. We don't want a variance. We want a bullseye result for the patient, because that is what we are here for. We don't want decreased precision or decreased accuracy in any way. So Six Sigma quality management is a data-based, customer-centered, advanced quality management model. The clinical application of Six Sigma quality management involves the combined use of quality requirements and laboratory performance to quantitatively evaluate whether a laboratory meets clinical testing standards. This evaluation is typically based on the expected defect rate. The ultimate goal of Six Sigma quality management is to implement laboratory risk management, as I told you earlier, that it identifies the areas of risk, and thus ensure patient safety through accurate results of sample analysis. Now, this was given by Guer et al., and the only reason that the last bit of it is in red and not in blue like the others is because that is what I have added to it, to remind myself that ultimately it is the patient, his result, his safety that we are all here for. So who is the customer for a medical diagnostic laboratory? the physician or the patient. And therefore, the tolerance limit is determined by either the physician or the patient, and also by the clinical impact of the number or the extent of errors. That is, how much will these errors affect the patient's treatment, his management, his course of disease, his recovery from disease, or his maintenance of health. Hence, the limit of tolerance is governed by the total allowable error derived from intra and inter-individual variations. Now, these are exercises that have been done globally for a very, very large number of data points. Using those data points, medical statisticians have derived what is acceptable total allowable error, what is the optimum, and what is the least that you must achieve. They have done the same for precision and for bias also. So when we talk about this total allowable error in our formula for sigma metrics, we are talking about this baseline which we are already provided. That is not something we have to calculate in our labs. It is further modulated by the bias. So limit of tolerance is governed by total allowable error and is then further modulated by the bias in the measurement and the coefficient of variation achieved. So all laboratory functions are directed towards better patient care, and hence these three variables need to be assessed in a standardized manner in the lab. And that is why we need to calculate sigma metrics for all processes and analysis. And I will try and show you how to do that. So we have the formula total allowable error minus bias, the whole thing divided by CV%, and that gives you the sigma metrics. It seems a very simple formula, but many of us who don't like maths, we have a mental block to using it. So what will we achieve by implementing sigma metrics in the medical diagnostic laboratory? It is supposed to help us improve quality, have confidence in our results, reduce our repeats and control runs and calibration frequencies, and thereby reduce the cost, and maybe also reduce the manpower required. So how do we apply? Methods of applying is the first thing is you, in any process, you have to identify first the method of the assay, and it has to be evaluated to see whether it meets the requirements for intended use. If I'm using a method for glucose estimation, that is your blood glucose estimation or your sugar estimation as is more commonly known, then I have to use a method that has already been set, that has already been validated, that has already been evaluated, and it tells me you can use this method from this level to this level. It gives you the linearity of the method, and it also tells me for what sample type I can use it, whether I can use it for all body fluids, including blood, urine, cerebrospinal fluid, or whether I can use it only for one or two of these. My next aim is to quantify the quality achieved for each analyte. When I say analyte, I mean in the medical jargon, I mean say something like hemoglobin, blood glucose, blood urea, serum creatinine, all these are analytes. Sigma metrics or efficiency of the assay can then be calculated once we have already achieved the, already calculated the, or derived rather, not just calculated, but derived the standard deviation, the bias, and the coefficient of variation. So areas or analytes that need improvement will then be identified. Our system quality control procedures can then be identified. New system quality control procedures can be identified and implemented, or changes in the system quality procedures can be identified. And then after implementing these new processes and procedures, you have to reassess what is the sigma metrics after implementing the corrective measure. And at the end of that, we should be able to reassure the customer that yes, our quality of service that we are providing has improved, and it is for the better of the patient, and it is what is required by the patient. You don't need probably any more than that. So we have Mr. Westcott, James Westcott, who is very well known in the medical field, in the medical laboratory field, because he has made life very simple for us by really going into the nitty-gritties of applying sigma metrics to the medical diagnostic laboratory. And he has given this drop down, which I'm sure you're finding very confusing. So let me take you step by step through it. It is quantifying the same process that I have enumerated here, but in a more, let's say, sequential manner. So first of all, let's go to this. We have to define the goals for intended use. That means we must assess that if I'm going to analyze this particular substance, I need to have a process that has a total allowable error of this, whatever that may be. Then for every analyte that is assayed, you know that the total allowable error will always differ because of the fact that there are two reasons for that. One is that the clinical implication of a unit change in the circulating concentration of an analyte is different for different analytes. If I have a 10% change in my blood glucose, it's not going to make any difference. But if I have a 10% change in my serum creatinine, well, that is very questionable. So therefore, the total allowable error differs for everything, for all analytes. Secondly, is that the analytical sensitivity of the methodologies that are available for measuring these analytes is different for different analytes, and sometimes there are several methods available for the same analyte, and their analytical characteristics may also differ. So having said this, having decided what we want to take and what is the total allowable error that we are going to consider, we go on to the next step, where we select the analytical examination process on basis of the total allowable error that we have assumed that we require. Having done that, we we validate the safety of this methodology or this examination process by calculating their CV and their bias. And CV is given by the standard deviation divided by the mean into 100 for percentage. And bias is given by the difference between the manufacturer's mean and our mean divided by the manufacturer's mean, again as a percent into 100. So once the method conforms to the requirements of intended use, you implement the analytical system. And that means you actually put into use this method for the measurement of either blood glucose or serum creatinine or hemoglobin. So how do you ascertain that the performance of your assay falls within the total allowable error? You can't just say that, okay, because you'll have to calculate something and then compare it. We don't calculate total allowable error in the lab. We say that this is the total allowable error given for this analyte. So you basically calculate the bias. So you run a sample of known concentration, for example, the control reference materials or a calibrator, and you compare the measured value with the true value as supplied by the company who is supplying the reference material. And the difference between the two, as you can see, is given in this chart, is the actual bias that you have. It may be a positive bias or it may be a negative bias depending upon what you have achieved. Once you calculate your mean bias and your bias percent, then you compare it to your total allowable error. So the allowable bias in any measuring system has to be less than 50% of the total allowable error. And that is when you can say, okay, now this assay is ready for use. So now coming back to the strategies here, what do? We know that the total allowable error is given by international standards, and it is given by Westcard also, and also by the CLI, that is the Clinical Laboratory Improvement Act, which was first started in 1988. Then you calculate the sigma metrics. So here we are calculating the sigma metrics because we are already given a total allowable error, and from our running of the controls on the assay we have derived the bias and we have derived the coefficient of variation. So we have all three components of the formula. Once we have done that, what Westcard has suggested is that you can select your QC tools and modulate them depending upon the sigma metrics that you achieve. Oh, sorry. And then you can design a new system of quality control if you want to, if you feel that is what is recommended. And he has gone through a very detailed exercise of actually telling us what we can do at what achievement level, at what sigma metrics level. So once you select a new design, you also do a risk analysis and you develop a total QC plan. You implement that plan and then measure its performance and monitor its failures. That is the most important. And once you've monitored the failures, you see whether or not your quality has improved. And then you go through the whole cycle all over again. If you have improved quality, well and good. If you have not, then you have to again assess where have you gone wrong and what do you need to do. So basically, I'm going to only talk about this one area, and that is selecting a QC tool as suggested by Westcott, and that would be the application of sigma metrics in a medical diagnostic laboratory. But before that, let me tell you, once you have got this and you have your Six Sigma, or rather you've got your sigma metrics calculated, you have this chart which has on the x-axis the observed inaccuracies, and on the y-axis it has the inaccuracies, and on the x-axis it has the observed imprecision. And once you have calculated there are areas on this that indicate what sigma metrics you have achieved. And these points in blue that are there are the various sigma metrics achieved by in different analytes in a certain company system. So what is 2 sigma is totally unacceptable. 3 is poor as per medical diagnostic laboratories. 4 has been considered as acceptable, it's just marginal. 5 is good, 6 is excellent, and more than 6 is world class. So this is the definition of this, or rather this is the gradation of sigma metrics in the medical diagnostic laboratory. Now let's talk about what Westcott has suggested. He has suggested corrections for several items in a routine laboratory on basis of the achieved sigma metrics. They may be either the number of controls required, or the frequency of the calibration, or the statistical rules to apply. I'm going to talk about the number of controls required. So this is the sigma metrics that we achieved in our laboratory for a few analytes. And as you can see, all of them are above 6. So now that we have achieved 6 sigma, how can we reduce our cost while maintaining quality is always a $1 million question. Westcott gives all these rules, and I know this is a very, very confusing slide. And all I want to tell you is that he has indicated that if you have reached 6 sigma, that is on the left, then the number of corrective actions you require are very minimal. Whereas if you are on the right hand side where there is 3 sigma, you have so many rules to apply. So let me just go to the next slide then. To be able to tell you what he means by changing our statistical rules applied, I have to tell you a little about the Levy-Jennings chart. If there are any people amongst you who are medical diagnostic people or even medical people, I'm sure you know what a Levy-Jennings chart is. It is a chart on which the daily quality control is plotted. The black line in the center is the mean that you achieve over a period of time, preferably at least a month or at least 20 points. The light green area is plus minus 1 standard deviation. Going beyond that is the yellow area, which is plus minus 2 standard deviations, and beyond that is the pink area, which is plus minus 3 standard deviations. Typically, in a medical diagnostic laboratory, we apply several rules. We say that if there is any quality control data point that goes beyond the 3 standard deviations, it is a straight rejection. You cannot use that run. You will have to reassess your process and see where things have gone wrong, and only then run the patient samples once you achieve a better control value. And there are others like 2.2s and 1.8x or 1.10x. 10x. So 10x rule is if if 10 consecutive points fall on one side of the mean, that indicates that your method has a bias. If you have 4-1s, that means 2 consecutive data points are separated by 4 standard deviations. So that means it is +2 and -2. So that also is a rejection rule. So there are many such rules. And which ones to apply and how to apply them is always a gray area, because there are no set rules for how you should apply them and which ones you should apply. So in this again, Mr. James Westcott and his son Sten Westcott have helped us a lot through this. So what we do is, what he says is, if your sigma matrix achieved is, as you can see in the left column, we have the sigma matrix from 6 down to 1 to 3. He tells you how many number of control runs you could run, how many number of controls per run should be used, and what are the QC rules that you need to apply. As you can see, with a 6 sigma, you need to run only 2 controls once a day, and the QC rule to be applied is just 1-3s. That means you are rejecting very few runs. There is no other QC rule that you need to apply. Practically, that means you are saving on controls, you are saving on the reagent to run those controls, and you are confident of your result. So once we apply this, since we achieved, if you go down to the 1 to 3 sigma, you can see that the number of controls to be run, the number of times it has to be run, and the number of QC rules that need to be applied are so much more, which means every second run probably would have to be rejected. So now let's talk about our scenario where we achieved more than 6 sigma in all those parameters. So what do? We change the control run. We were running 2 control levels twice a day, and now we made it 1 control level, or rather 2 control levels once a day. So actually, I know on the right hand side I've given you the number of control aliquots that are saved, but what I actually mean there is, when I say 60 aliquots, that means 60 aliquots in the month, but in each aliquot, you can do at least 10 to 15 or maybe 20 analytes. So in effect, we are, for all the analytes listed on the left-hand side, the total would be only 60 aliquots of saving. So earlier we were using 120 and we have brought it down to half. That means we are saving in the cost of the control and the cost of the reagent required to run those controls. So that is quite a lot of saving, and yet our quality is being maintained. So based on these advantages, as reported as well as demonstrated, every diagnostic setup must implement or should implement sigma metrics as a quality improvement tool, not only to improve quality, but to reduce cost and yet have confidence in your results. That is one of the main things. Now let's talk about calculating sigma metrics in various phases of the medical diagnostic laboratory. So far, I have been talking about only one small area of a medical diagnostic laboratory where the sample is actually being analyzed for the particular analyte that we want or that has been asked for. The sample, we are assessing glucose in that, or we're assessing total protein in that, or bilirubin in that. But there are many things that happen before that phase comes and many things that happen after that phase comes. So typically a laboratory is divided into 3 phases: the pre-analytical, the analytical, and the post-analytical, which is given diagrammatically in the image here. Basically, in the pre-analytical phase, we have many steps. First, the investigation is ordered by the doctor, then the bill is generated, then the tubes are prepared, then the sample is drawn by the phlebotomist or collected if it's a urine sample. The sample is then sent to the lab. It is received there and processed. For example, it has to be centrifuged or it has to be kept aside for some time. Then equipment daily maintenance is already in progress, hopefully, by the time the samples reach there. And then we have to validate and verify processes through the measurement of internal quality control and external quality control. Those of you who are medical people will understand this, and those of you who are not, I'll come to it a bit later. In the analytical phase, we have just one step. The sample is analyzed for the requisite analyte. In the post-analytic phase, we again have validation of results and release of reports. So in each process, there can be you can calculate sigma, or rather you need to calculate sigma, and I'll tell you how you can do it. So what are the pre-analytical phase? What are the things that you could actually consider as indicators of quality in your pre-analytical phase? So every lab has its own list of reasons for rejecting a sample. It may be that the sample is too little, or that it has been brought at the wrong temperature, or that it has been stored overnight and cannot be used for many reasons, or that it has come in the wrong tube. So number of samples that have been rejected could be a quality indicator. Number of samples that are hemolyzed, which will be evident mostly after centrifugation of the sample. Number of samples that have been received in inappropriate containers, or that have been transported without the appropriate temperature monitoring. For example, there are some analytes that are stable only if you transfer the sample on dry ice or at 0 degrees. And for example, estimation of methotrexate, estimation of interleukin 6, which all of you must have heard of in the COVID times. Everyone needed that, but those results were reliable only if the sample was transported on ice within an hour to the lab. That is, so there can be many reasons to reject, and there can be many areas that we can use as quality indicators. So then how do we calculate sigma metrics for that? It's very simple, actually. In the analytical phase, it may seem more mathematical and more complex. But in this pre-analytical and the analytical phase, it's very simple. The calculation is simple. You'd say the number of samples that have been rejected over the total number of samples that have been received in the same period of time that you have taken the samples rejected. And you multiply that by 1,000,000, you get your number of samples rejected per million samples received, which means per million opportunities. Similarly, if you want to do about number of samples hemolyzed, you just change the numerator of this equation, or rather of this calculation, and put their number of samples hemolyzed. So it's very simple, and you can use these as your quality indicators, and they can be expressed as sigma metrics achieved. And you can also decide that in my laboratory, because of the type of patients that I'm dealing with, because of the type of sample and the number of samples that I receive, because of the size of my lab, I need to achieve at least this much sigma metrics in this area. In each quality indicator, you can set your acceptable limits. And that is what is meant about quality management system. Similarly, in the post-analytical phase, you have the number of times critical results are not conveyed in the specified time. For example, if you know, all of you must be knowing what high sensitivity troponin I is. It is used on a patient who comes to the emergency unit with chest pain. And you want to know whether he actually is having a heart attack, or is it just something non-specific. One of the best tests for that is HS-drop-I. But since this is such a critical situation for the patient, because a decision has to be made whether or not he has to be admitted as a case of acute myocardial infarction, or maybe discharged as just a non-specific gaseous distension, you have to make sure that the result reaches the clinician or whoever is in the emergency unit looking after this within that 1 hour that is required. In case of HS-drop-I, it is 1 hour. If your results are not conveyed within 1 hour, then you have to specify the number of times that has happened over the number of times that it was not supposed to happen. So that is your number of opportunities that you had. That means the number of critical results you generated and number of times the critical results were not conveyed. When you give a ratio of that and multiply it by 1,000,000, you'll get your sigma metrics of critical results. Similarly, number of times a clinically effective turnaround time is exceeded. So clinically effective also, and what you have claimed also. Both these things are important. We in our lab say that we give our results within 2 to 4 hours for a routine analyte, say for blood sugar estimation, blood glucose estimation. If we are not able to give it in 4 hours, that becomes a non-conformance. And that will be counted as one of the times that the clinically effective TAT was exceeded, or the assigned TAT was exceeded. So that you can use again for sigma metrics of your turnaround time. Then actually turnaround, let me just define turnaround time. Typically, if you take a holistic approach, turnaround time is the time from the time that the doctor orders the investigation to the time that he receives the report. But practically in laboratories, that often is not the turnaround time. We take it from the time that we receive the sample till the time that we release the result to the doctor. Then where their analyzers are not connected to a lab information system, then all the results have to be transcribed onto the report. So there could be a lot of transcription errors. So all of these can be used to calculate sigma metrics. They can be all used as quality improvement tools, that you have achieved these many transcription errors. Now you have to reduce them, and this is how you're going to do it. You set a system, you re-evaluate the system after some time, say after whatever, say 3 months, 2 months, 1 month. And then you say, okay, I have achieved an improvement, or I have not achieved an improvement. I need to do something differently. That is what it is all about. Applying sigma metrics in a quality system for quality improvement. Otherwise, if we do not use any methods to improve, then it's pointless calculating the sigma metrics. As in case of pre-analytical errors, these can be calculated to give the errors per million opportunities. This I've already told you, that in addition to calculating, we must set acceptable limits for each pre-analytical and each post-analytical parameter. And they can be used as key quality indicators. Now, one more consideration that is there. So we talk about, even though we say that even 4 sigma metrics, achieving 4 sigma in a medical diagnostic laboratory is acceptable. But there are a few considerations that go in favor of achieving 6 sigma rather than just stopping at 4. And the reason is that the shift in the mean with the new lot of controls, or with any other thing, happens all the time, as I've already told you, because of natural sources of variation and unexpected errors and the inherent variation in the system. And this may cause a sample result to be out also. So energy, time, and expenses are incurred to achieve acceptable results if the process is not 6 sigma. 6 sigma tolerates a shift in mean up to 1.5 standard deviations, which I've already told you. Actually, I haven't shown you that slide. I wish I did, but anyway, it doesn't matter. While as 3 sigma tolerates less than 1 standard deviation, a shift of less than 1 standard deviation. So therefore, you can understand why it would be important to save that energy, time, and expense by achieving 6 sigma rather than 3 sigma. And hence changing from 3 to 6 is definitely advisable. So then what is it? There was one laboratory that applied 6 sigma metrics, calculated sigma metrics, and then altered their QC rules. And the advantages that they counted, they actually assessed their whole process in terms of commercial as well as in terms of results attained. So they found that the results became more accurate and precise. 70% reduction in repeats was achieved. 50% reduction in quality control failures was also achieved. Then they also achieved a reduction in the required troubleshooting, obviously, and a reduction in the turnaround time, which of course goes favorably for the patient. and a 45% savings in supplies for running calibrations and controls. Now, saving on commercials and infrastructure without compromising quality and, in fact, improving quality. That is the whole crux of the matter. That is why we need to apply Sigma metrics to our systems and apply it well, apply it with a proper thought process. And when I come to the paradoxes, you will understand what I mean by that. So now let's come to the paradoxes. In these, I have three main concerns. One is the components of the formula and the blanket statement that Six Sigma is the best. Then is the different practical connotations of achieving Six Sigma in different sizes of laboratories. Does one glove really fit all? And then the method of improving Sigma. So I will address each one of these paradoxes one by one, and I will try and explain to the best of my ability. But before I do that, let me tell you about a simple limitation of Sigma metrics, and that is in its bringing it from theory to practice. Once you have achieved Sigma, what next? Of course, this I'll be talking about, and inherent limitations. But from theory to practice is one of the most difficult parts. All of us read about Six Sigma. Many of us are trained in Six Sigma. Many of us have adopted Six Sigma or other Sigma metrics and the Sigma metrics way of quality control and quality management. But the bottom line is that we are not very savvy with numbers. We are medical people and we have done less of maths, even though statistics is part of our course. And we are deterred by the thought that there is a lot of statistical calculations required. But what I want you to know is that that is not so. Please get rid of that deterrent. Actually, it's very easy and there are no direct calculations at all that are required. You just have to set up your Excel sheet once with inbuilt calculations, and then every month you just put the month's mean and standard deviation and coefficient of variation, and you're done. You get your Sigma metrics automatically. And that Excel sheet you have to, in any case, keep. It is requirement of NABL 112 as per ISO 15189 2022, which is the international standard for accreditation of medical and testing laboratories. So if you have to do that, why not? You might as well do it. And it's very simple. So this is all you need to do. You have these 1, 2, 3, 4, 5, 6, 7 columns. In the first left column, you list the analyte. In the next column, you list the manufacturer's mean. In the third column, you list your cumulative mean. I have used cumulative mean so that you go over several months of the control values and you take the cumulative mean rather than of 1 month or 1 day or whatever. Then you list the cumulative standard deviation and the cumulative coefficient of variation, which these equipments that you have, these automated analyzers or even the semi-automated analyzers, they automatically calculate for you. You don't have to do any calculations. You have to just pick it up and transcribe it. Yes, please be careful, no transcription errors. Now, the difference between your second and third columns, that is the manufacturer's mean and the cumulative mean, gives you your bias. Total allowable error is the last one. And that is something that is already given. You don't have to change that. It is, therefore, you can add one more column here now on the right-hand side, and that would be the Sigma matrix. And in that, you can just write the calculation. Column number 7 minus column number 6 divided by column number 5. And you're done. You have your Sigma metrics in front of you. You don't have to do anything else. It is not so difficult. It is very easy. So open up your mind to it. So now let me talk about the first paradox that I have talked of. I have enumerated the components of the formula. So we have calculation of Sigma, We know it is total allowable error minus bias divided by the CV%. Therefore, obviously, a sigma achieved will be directly proportional to the total allowable error. The greater the total allowable error, the better is the sigma that we achieve. But would that be nice? I don't think we would want to have a very high total allowable error. So in 2014, there was a strategic conference on quality specifications in Milan where they discussed a lot about whether to use measurement uncertainty or total allowable error. And they still haven't come to a conclusion. So actually, it's futile telling us anyone this because there is no consensus about it as yet. So I have already told you that total allowable error has been given by the Clinical Laboratory Improvement Act of 1988. But I'm sure you can understand that it is decades old. Should we be using that? And actually, that was developed as not a measure of total allowable error. It was total allowable error meant for proficiency testing assessment. I think I'll have to tell you just one quick word about what we mean by proficiency testing. In a medical laboratory, the quality management system, apart from all the other parts, has two major areas. One is the internal quality control that helps you in your daily runs and that you assess your systems by on a daily basis, which gives you the precision and to some extent some bias or some accuracy, but not all of it. While as external quality assessment or proficiency testing is through a sample that is sent to you by a PT provider, so-called proficiency testing provider, who is a certified provider, and they do not give you the actual result expected, but they take your results and they collate it along with thousands of the results from thousands of other laboratories, and they give you a histogram and several statistical evaluations of as to where you stand in the larger picture. How much of bias do you have? Do you need to do anything? Is your methodology acceptable or not? So in a proficiency testing, the total allowable error will always be higher than in the tight confines of your single laboratory environment. So therefore, using these forms of the total allowable error is not right. Then in 2019, CLIA has given new acceptable limits for total allowable error, but again, they are actually for proficiency testing. And but they're also similar to the 2014 specifications given by Ricos and Carmen, who have actually calculated the intra-individual and inter-individual variations that I was talking about. And therefore, you must be very careful, even if the CLIA 2019 and the Ricos-Carmen charts give you a total allowable error that is much less than what CLIA 88 gave you. It is CLIA 2019 or Ricos-Carmen that you should be using to be correct to actually correctly calculate your sigma metrics. So that is about the total allowable error, that component of the formula. And here you see the acceptable performance proposed by CLIA 2019 and the total allowable error as per Ricos and Carmen. Even in that, you see how much of a difference there is. And that is because the acceptable performance proposed by CLIA is for proficiency testing, while as total allowable error given by Ricos and Carmen is for within your lab. So therefore, it is the right-hand side column, the values given in Ricos and Carmen, that should be ideally used as total allowable error in your calculation of sigma metrics. I want to give you an example. I've already talked about high sensitivity cardiac troponin I. And let us say that earlier, CLIA used to talk about, actually this, when troponin was started, CLIA gave the acceptable limits as plus minus 30%, the total allowable error of plus minus 30%. Now, if you look at, suppose we have a 40-year-old male patient who comes to our emergency department with chest pain and his HS tropi is reported as 37.5. Now, if you look at the biological reference interval for males, 28.9 to 39.2 is the normal. So 37.5 would be in the normal range. But if your total allowable error or your total variation acceptable is 30%, plus minus 30%, then his report could actually be anywhere between 26.25 to 48.75. So therefore, having such a high variation would significantly increase the false positives as well as the false negative predictive values. And that is why for such analytes, we need much tighter allowable variations. And therefore, the acceptable performance for PT should not be the total allowable error for calculating sigma. So actually, for HS tropi, they haven't yet given any. They have just recently given. And did I list it here? No, I didn't. So I think it is 10% that has been given now. So once it is 10%, it is acceptable and usable, and we can take a decision in the emergency room based on that report. Otherwise, doing a test is futile if you cannot take a decision on that. Now, let me give you another example. Suppose you have a bias of 4% in any system that you are. In any assaying systems, and the coefficient of variation achieved is 3%. So if you take a total allowable error of 30% as may be listed by CLIA, the sigma you have achieved is 8.6. That's fantastic. And if you reduce the total allowable error to 25%, your sigma also reduces to 7. And if you reduce the total allowable error further to 20%, the sigma achieved will be 5.3. That is but natural because total allowable error is the numerator of your calculation for sigma metrics. So as your total allowable error reduces, so will your sigma achieved reduce. Now with a total allowable error of 20%, now, we are saying that what the 20% that has been given by Ricos and Carmen is a better measure. So let us take that and presume that we have achieved only 5.3 sigma. Now, with a total allowable error of 20%, if you reduce your bias to 2%, then the sigma achieved is 6. So therefore, a sigma of 6, which is a lower numerical value, with a total allowable error of 20% and a bias of 2% is much better than a sigma of 8.6. 8.6 is much more than 6. But there the total allowable error that we used was 30% and the bias was 4%. So what I'm trying to say here is that each component of the formula is important. Picking up the correct total allowable error from data provided is important. Achieving the lowest bias and the lowest coefficient of variation is equally important. And that is where the paradox arises if you do not use the correct values. And that is why I call this a paradox. So calculation, same thing as calculation of sigma would be sigma achieved would be directly proportional or rather indirectly proportional to the coefficient of variation. And currently, and maybe in the future too, better and better methods with more stable methodologies are being used and manufacturers have reduced the CVs drastically. So therefore, our CVs are reducing, and since the CVs reduce, the sigma will be higher. And then also, hopefully, in the future, we'll have standardization and harmonization of all methodologies for many analytes. And that is a relief. At least we have some hope in the future that we will not be probably having to deal with a lower CV or a lower sigma, I mean. What would be the future scenario? The decision limits of an analyte. So today we know that I can decide that I will decide to change the treatment of a patient on the basis of a urinary protein being, say, 1 gram and going from 1 gram to 2 gram. In the future, it may be going from 1 milligram to 2 milligram. That requires an alteration in management. So therefore, your decision limits of analytes for medical intervention or alteration of management might change in the future. They might become more rigorous. And then what will happen? Then technology may, at the same time, technology will also improve and methodologies will also allow for less error. So therefore, if our total allowable error reduces, so will our coefficient of variation and bias. And therefore, probably we will automatically have the same sigma matrix, and yet that sigma matrix might even indicate a better performance than before without even changing the sigma expectations. I hope that's understandable. Now let's go to the next paradox. The different practical connotations of achieving Six Sigma in different sizes of laboratories. Does one glove really fit all? We all have different hand sizes, different feet sizes, different shoe sizes. So therefore, how can one glove fit all? But applying this to Sigma, I would like to say a few things. Supposing you have achieved Six Sigma, I'll come to this first. What is next? So it has been said that total quality management is a journey. It's not a destination. You have to continually improve. And that is what your quality management is all about. It is about improving consistently and not stopping at any one destination. So Six Sigma is not a destination. You have to go beyond that also if you can. Six Sigma equates to the absence of customer perceptible defects. So now again, we in the laboratory know the nitty-gritties of our assays, but the clinician and the patient do not know. And what they perceive as a defect may be much less than what we perceive as defects. So are we going to continue with this definition that Six Sigma equates to the absence of perceptible defects? Is that what we are really striving for? And suppose we do achieve Six Sigma, what next? What, if any, is the significance of achieving beyond Six Sigma? So let me give you, suppose you've achieved Six Sigma. Now, before you come to the examples and all, let me tell you, you must be very skeptical about it. There are many questions that come to mind. If Six Sigma is world class, then what is 20 Sigma? As you can see in my list of Sigma, we achieved 20 and even 23 and 25 Sigma. But what does that signify? If a method is 18 Sigma, does that mean it is 3 times better than the Six Sigma? If a method is that good, are there any practical benefits to achieving higher than Six Sigma? Or is this just surplus excellence? So first and foremost, you should be very skeptical, your first reaction. So you must check whether the method is really performing at Six Sigma. Is it that just once it has achieved Six Sigma, or is it that it is consistently performing well? Is it performing Six Sigma for all levels of the controls? Like for each analyte, we put up 2 to 3 levels of controls, low, normal, high, like that. So is it performing Six Sigma in all these levels? Or is it just a fluke, just this once, and we just get excited about it unnecessarily? So what do we need to do? We need to check the sigma of all the control levels. We need to see if Six Sigma is achieved only in one control or even in less than 2 out of 3 control levels. We have to then, in that case, accept the lowest sigma achieved as depictive for that analyte. If Six Sigma is achieved in all levels of controls in the latest sigma calculation only, then you must observe for another few cycles of assessment. If Six Sigma is achieved in all level of controls over a period of more than 3 months, then you can look at Westgaard's suggestions. As I told you, he has been our go-to person for quality control. We need to go look at his suggestions regarding the number of controls to be used, the frequency of putting up controls, the QC rules to apply, the frequency of calibration, the number of standard deviations that we can assign as acceptable. And then how do we quantify beyond 6 sigma? So 6 sigma is supposed to be 3.4 defects per million opportunities. And I have got this off the net. So 7 sigma, what I'm talking about is off the net. 7 sigma is supposed to be 0.020 defects per million opportunities or 2 defects per 100 million opportunities. 7.5 is similarly 1 defect per 1,000 million opportunities. 7.86 sigma is 1 defect per 10,000 million opportunities. 8.53 sigma is 1 defect per 10 raised power 12 opportunities. That is a million opportunities. So is this practically feasible and is it really required? Both these questions have to be answered, and this cannot be answered directly by any one person. It has to be a practice in your laboratory, an assessment of what are the number of samples you're receiving, what are the number of types of tests you're doing. I'll come to that soon. So what are we doing? If we go from 5 sigma, which is 230 defects per million opportunities, or 23,000 defects per 100 million opportunities, and 6 sigma is 3.4 defects or 340 defects per 100 million opportunities, so that we have the same denominator and an easy calculator. So if we go from 5 to 6 sigma, we are going from 23,000 defects per 100 million opportunities to 340 defects per 100 million opportunities, which converts to a reduction by a factor of 68. I'm sorry, please bear with me. Just a few calculations to put my point across. Then if we go from 6 sigma to 7 sigma, what are we achieving? We are going from 340 defects per 100 million opportunities to 2.
I'm sorry, I think Santoshji, I think there's some problem with her connection.
Yes, sir.
That is a
Yes, ma'am, please continue. Today's information is very good.
Are we doing enough calibrations? Can the clinician really make a decision on a patient management with this low precision? We have to weigh all these things before we take a decision. So now coming to what next, and does one glove fit all? Let me tell you about that. I'll give you an example. Who is our ultimate customer? The patient? Let me skip this slide. I think it's too much of calculation just now. So let's talk about a small standalone laboratory. Let us presume that the lab receives 100 samples every day and approximately 5 analyte estimations per sample are done. So therefore, every day, the number of tests that will be done in that laboratory will be 500. So therefore, there are 500 opportunities for mistakes or for errors in the analytical phase. Number of tests would be 15,000. Number of tests per year would be 180,000, which would mean 0.18 million. So if we achieve 5 sigma, then the number of defects would be approximately 41 per year, applying the fact that 230 defects per million opportunities is what 5 sigma signifies. Improving this to 6 sigma would bring it up to less than 1 defect per year. Now, improving to 7 sigma? My God, that would be less than 1 defect in 140 years. I have not shown you all the calculations, but I have gone serially as per the assigned defects to every sigma and calculated according to this sample load of this laboratory. So is this improvement justified at the extra cost it is most likely to incur? Probably not. Now let's take a large hospital laboratory, and let us take that the number of samples per day is 2000, and accordingly the number of samples estimated, the number of estimations per year would be 3.6 million. Achieving 6 sigma would mean that we have 3.4 into 3.6 defects per year, which is equal to 12.24 defects per year, or approximately 1 defect per month. Can you imagine that? Do you think that is acceptable to any hospital or doctor? Do you think that they would be happy, or they would accept the fact that they may mismanage one patient every month because of one laboratory error? I don't think so. I don't think that is right. So again, in this very large hospital laboratory, if we improve from 6 to 7 sigma, we have 1 over 140 defects per month. That would mean 12 over 140 defects per year, which would mean less than 1 defect in 10 years. Is this improvement justified? This seems to be an overkill. But any improvement from that 6 sigma where we are making one mistake every month, definitely we have to improve on that. We may not need to reach 7 sigma, but we, yes, have to improve even if it costs us. And is there any improvement in reputation? I'm sure, quite likely. But are we saving more lives or improving management? Yes, we are. So therefore, it is quite likely that we will have to incur that cost and make this improvement. Maybe not up to 7 sigma, but maybe 6.2 or 6.3 or whatever it calculates up to. Now, what are the methods used for improving sigma? And that comes to the last paradox that I have. So in a medical diagnostic laboratory, the commercial considerations, if we talk about them, I've just told you about, should we be achieving, what do we achieve by shifting from 5 sigma to 6 sigma? A defect rate shift from 1 per month to less than 1 in 10 years. Do we really need that? Okay. What extra do we spend to achieve this? Now, the point is that we may say that we will have increased use of calibrators and increased use of controls and therefore extra expenditures. But how much would that really be? So let's calculate. Supposing the control schedule is altered minimally from 2 levels run twice daily to 3 levels run twice daily, as suggested by James Westgard. Hence, every day the lab would be using 6 aliquots of the reference material instead of 4. Straight away, that means a 50% increase in the cost of the reference material alone, to say nothing of the cost of the reagents used to analyze that reference material. The question that arises here is that by this minimal change in control schedule, will our sigma matrix actually increase from 6 to 7? Probably not. To achieve a 7 sigma, we would probably have to alter the calibration schedule as well. And this could also mean that we would require more personnel. Now, all this increase in expense incurred may not justify the advantages of an increase in sigma metrics from 6 to 7 in a small laboratory, but definitely, probably, I would say yes, in a large laboratory, it would be advisable because you cannot live with 1 defect per month. And we haven't even considered beyond 7 sigma yet. So to conclude, I'd like to say that I'd just like to bring out a few bullet points that evolving times mandate evolution in our assessment of assay performance. Sigma metrics calculations offer a quantitative method for this. While calculating sigma, one must use total allowable error and not acceptable performance of PT, as I have detailed earlier. Effective corrective measures for assays with low sigma need to be implemented. And I must tell you, as of now, there is no consensus and there are not enough data from all over the world to say that yes, correcting or changing your schedule from this control schedule to this control schedule is going to improve your sigma by this much percent if your sample load is this much. There is no bottom line, there is no baseline to go by, and we have to create a baseline, and we have to create a baseline that will have to be done by statisticians because they're good at maths. They will have to take into consideration not only the actual utilization of controls and calibrators, but also the baseline of the number of tests or the number of opportunities that that particular lab has in 1 year. The opportunities for defects, I mean. Then one must also remember that the clinical efficacy is ultimately of prime importance, and overachieving a high sigma may be unnecessary and uneconomical. The decision limits for medical intervention are also changing, and hence interpretation of performance sigma may also change in the future. Be prepared for it. But as I told you, our methodologies will also improve. So ultimately, what we calculate today as 6 sigma might still be the same 6 sigma next year, but might indicate an even better performance. Improving technology allows for newer methods with lower bias and lower CV% so that performance may improve without alteration in sigma, as I just said. To conclude, I'd like to quote McAdams. He says that the data you can get from a good Six Sigma program and good projects can help you focus a business strategy. But no, it is not the end-all solution to saving a business. You have to do more than that. You have to apply it with consideration of all your other parameters and see where it will benefit you and where it is not beneficial, where achieving 6 is important or more than 6 is important, and where achieving 4.5 is adequate. That is my bottom line. And I hope I have expressed my reservations, and I hope none of you are in this state. But thank you very much for your patient listening. And I would like to show you a picture of my hospital. This is where I work. Thank you very much. I'll stop sharing now.
Thank you very much, ma'am. It was a very, it is one of the best presentation as well as the explanation which we have since last more than 2 years. We are doing these programs, so it is one of the best program. I would like to request all the participants to kindly switch on their video and switch off their mics so that we can take the pictures. And you can also.
I'm a bit technologically challenged, and I'm not able to. I'm not able to unshare.
Santoshi, can we do this?
Let me try from here.
Yes, sir. Yes, sir. No problem, ma'am. He will do that. Don't worry. Ma'am, now there are 2-3 things which I would like to request you to ask. One is, ma'am, this CLIA, the Clinical Laboratory Improvement Amendment 88. It is, I think it is of US. It is basically in India.
Yes.
We are also complying the same standards as are in US.
It doesn't really matter whether a lab is in the US or whether it is in India. The patient is the same. Okay. And achieving quality is the aim of every laboratory. Achieving a good quality, effective quality towards better patient care, towards better management of the patient is very important. So whether it is made in tomorrow with Modi ji's very good, he has brought India to a very different level. And today we are following the West. Tomorrow the West may follow us. They're already following us in yoga, so maybe they'll follow us in this also. Very good. But I think this is the best standard which we have right now for the whole world. Yes, but it is the CLIA is a proficiency testing standard. It does not give you the total allowable error. It gives you the limits of acceptable proficiency testing assessment. While Ricos and Carmen are those who give you the actual total allowable error. And we should use that for the calculations.
Okay. But it was very good because we were not aware. Actually, being a meteorologist, I was not aware of that. And certainly it opens our eyes. Certainly it is a very good exercise, ma'am. And we would like to request you to come once again and give us, enlighten us about the new experiences, whatsoever you have. Ma'am, one another thing, you must be having some Excel sheets where you must be doing the calculations. Am I right?
Yes.
If you kindly share these Excel sheets, we can share it to all the participants, and these Excel sheets may be used in all the laboratories wherever required. If you feel it is not
Every laboratory that is NABL accredited has the Excel sheet. They have it ready because they have to do it. It is
okay,
mandated.
but
But
ma'am,
those who are not, I will definitely share that. I
very
will
good.
happily
Because
share
ma'am,
that.
we have, I hope that we have about 5 lakhs laboratories in the country, medical laboratories which are doing the testing.
That's an underestimation. We probably have many more than that.
Ah. So, ma'am, it will be a very good exercise if we provide them the Excel sheets, how to do the testing, how to do the calculations.
Wawa Timura. Remember movie? Wawa Timura.
I had actually put in the
Me,
a mode
My
of
request is,
actually going through
to
it,
kindly
but
switch off your
I couldn't
mic.
get that.
Mr. Moidin, kindly switch off your mic, please, if you're not asking any question. I'm sorry, ma'am, please.
Yeah, what I was saying is that I had actually, in my first presentation, I had put in a way of taking this straight to my Excel sheet so that you could see how it is done. But I was not able to do it just now. I'm a bit
No issue, no
technologically
issue. So,
challenged
ma'am,
in
I will
that.
take your Excel sheets. I will take your Excel sheets and will share to all the participants. Ma'am, second
Sure.
is your presentation. I would like to share your presentation on our all portals. We have Facebook, we have this Twitter and Instagram, every portal and YouTube. And if you agree, this presentation, what you have given today, we will put it on our all channels so that anybody at any point of time can see, can understand, can, if you permit.
I will be honored.
Thank you. Thank you very much.
I will send you the presentation, or do you already have it recorded, I think?
Ma'am, recording we will put, but presentation will be very good if you kindly send the presentation.
I will send it. I will happily send it.
Thank you, ma'am. Anybody who want to ask, I think a few of the people are raising their hands. They can ask the question directly, please. I think few of them from outside the country, they want to ask. Dikshit sir, you want to say something? Dikshit sir? Santosh ji?
Ji,
Ji sir, please sir, please.
no question. This is honor that madam has come here and given the good presentation to us, and we got enlightened. And this was the new topic, and we are very very thankful to her, and we congratulate her also for such presentation.
Thank you,
Thanks,
sir.
madam.
Thank you.
Thanks at all, sir.
Thank you, sir. So actually, the purpose is how basically Six Sigma is very common for the industries. How to do in the medical side, it is a, eye-opening actually, even for me.
So no, medical science, it is happening from since long time
All
that
of
I
you
am
in
learning.
between. I'm
But
sorry I missed a few. Can you please, if there is a question, can you please repeat it?
Anybody who want to ask the question?
Yes,
Yeah,
I have
please,
one question.
please.
Yes, please.
Actually, thank you for this great session, ma'am. As you told that we have to improve from 6 sigma to 7 sigma. So I just want to know that is any hospital or laboratory in our country or in a worldwide who can implementing those kind of things in actually.
Yes, as I told you that it is not just about improving from 6 to 7 sigma. It may be good for some labs, it may not be required in other labs. It is what I'm trying to tell you is that sigma alone should not be the reason. The calculation of sigma alone should not be what your quality system improvements is based on. Yes, you can use it to see whether or not you have improved. You can use it to see, okay, do I need to achieve more? But as I showed you a list of analytes which we had done and in which we were achieving 18 sigma and 23 sigma, and we have achieved that. And we were using the correct total allowable error. So we went through that whole exercise to check. That is what got us very thinking, okay, what is wrong? Have we done something wrong? How can we be achieving that? But it just so happens that the that the equipment that we have is highly sophisticated equipment. The reagents provided have very low coefficients of variation, very minimal, and probably our environment is also well maintained. So therefore, we might be achieving it, but it's not a consistent achievement. I have to say that also. But at that one point of time, we did achieve it, and having implemented some corrections, we didn't need to go beyond 6. Why would we go beyond 6? That is an overkill. I actually haven't given you many examples that I had. Maybe sometime next I will talk only about examples, real life, real time examples about how it made a difference in creatinine. What did I do for that? What did I need to do, and what seemed to be the wrong thing to do? And for several analytes, we have gone, I have a few case examples, but I didn't put them on today. I thought it's a lot getting this much.
Thank you. Thank you very much, ma'am. Thank you. So, ma'am, there's one another question. What is the concept of sigma metric in quality control, and how does it relate to process capability?
Process capability can be measured in many ways. Sigma metrics is one of them. It is a quantification of that quality process. So when you say, okay, I made this mistake here and I'm not able to quantify it, then how can I make an improvement and say, yes, I made this improvement. Yes, I have improved. So when I apply sigma metrics and take each numerator and denominator and multiply it correctly and divide it correctly, once I have quantified what I'm achieving, then I make a change in my system, quality system, and then I recalculate what I have achieved after the improvement, so-called improvement that I have implemented, and see that has it actually resulted in an improvement. That is what sigma metrics helps us in quantifying it, giving it a numerical value. Just saying that we are better doesn't help, but saying that yes, we went from 4.2 sigma to 4.6 sigma, that has been an achievement that you can relate in terms of numbers.
Thank you. Thank you very much, ma'am. I understand that today you are traveling outside the country, and even after having a lot of other things to do, you have taken the time. Really, I appreciate. I'm thankful to you. I'm grateful for this presentation. Thank you very much. Thank you all. We will meet next week.
Thank you so much.
Thank you very much. Thank you, sir. Thank you, ma'am. Thank you.